everyone to this webinar. You may be wondering about the title, Polar Bears and Populate. It's a bit of an odd title, I'll give you that. But the reason we chose that title is really, it's about diversity and courses, or horses for courses, so to speak. So, you know, what we're looking at here as part of this talk is to try to get a message across about diversity and how important it is to have the right microbes in the right environment to do the right job. So I'll get started with that. So there'll be a little bit about soil microbiology, because I can't really talk about this if I don't give you guys a little bit of a background about soil microbiology and what happens there. So look, plants and these soil microbes, and particularly fungi and bacteria, but also some other things like protozoas, have a very, very tight interaction. They actually really live together and have for hundreds of millions of years. And so they're present everywhere in the soil, these microbes, but they tend to congregate around the area of the root. So it's called the rhizosphere. So they form these really tight, mutualistic, symbiotic relationships. So what it really is, it's a relationship where both the microbe and the plant gets a benefit. So when you think about that, it's a bit like a honeybee and a flower, or a cleaner fish and a shark. So if you think about that, then you realize that in order for this to happen, the plants do their part and the microbes do theirs. So, you know, it's that idea that the plant feeds the fungi with sugars and amino acids, and the fungi then gives the plant things like phosphate and the like. So normally this is about an hour and a half talk, and I've had to condense it down a little bit. So if you guys are interested in any of the topics I'm raising, please feel free to you know, put it in the Q&A or send an email. Now, this is a bit of an overview about the sort of things that you can expect when you have really active and a diverse population of microbes in the soil. So you do things like cycle nutrients. So, you know, very, very rapid turnover of nutrients. The leaf litter falls down, it gets broken down, the material gets released into the soil, it gets taken up by the plants, and it's a cycle. So you got things like that. Then we have things involved more in the solubilization of specific nutrients. So we're looking at things like phosphate, which is often tied up, increasing available carbon dioxide for plant growth. So when microbes break down that mulch or that compost you're putting down, that increases the CO2 around the plant, and the plants feed upon that. There's this idea of being able to fix nitrogen. So nitrogen, as you know, is one of the 3 macronutrients that everybody talks about. So a lot of microbes have the ability to do that. You may be familiar with things like legumes, so things like your clovers, your peas, your beans, how they have those nodules on the roots. That's where nitrogen fixing occurs. But there's also a whole range of bacteria that don't associate with the plant, that are free living in the soil. They also fix some of this nitrogen. Then there's disease protection. That's a really, really vital aspect that microbes play for plants. So they do that by competing. So they take up the space that you would normally have. For example, a pathogen could occupy a space, but if there's lots of beneficial microbes, they'll drive that pathogen out. And then there's direct inhibitions, where these microbes secrete compounds to, in effect, kill or inhibit the growth of pathogens. And you'll see some slides on that a little bit later. There's obviously the idea that you can stimulate plant defenses. Microbes also do that. And lastly, but perhaps not least, is the idea of plant growth hormones. So things like auxins, which you find, and cytokines, which you find in rooting powders, and gibberellic acids, which are involved in really determining sort of the shape and the internode length, so the length between where leaves come out on a plant. So there's enormous diversity in the soil. You'll see a little bit of a slide here of what we do at Neutrog. We get soil samples in, we mix them with saline, and then we start to look for beneficial microbes. So there can be about 30,000 different species of bacteria in any specific soil type. And that variation depends upon the type of soil and the plants growing there. And obviously also the environmental conditions, dry versus hot, wet versus wet versus dry, and also obviously hot versus cold. So some areas are really dominated by fungi and others by bacteria. So if you've got a lot of trees in your garden and large shrubs, that tends to be dominated by fungi. If you've got a very lush lawn, it's dominated by bacteria. So like I said, there's a lot of variation. Most of these microbes are really poorly described, and we can only grow a certain limited number in the lab. But what we do know is that, you know, we do need this interaction. We just don't know what all the interactions are. So diversity is essential. So if you're applying a product, you should be getting as many microbes in there as possible, okay? And that's why the ethos at Neutrog is diversity, diversity, diversity. If you take nothing else away, think about diversity as being so critical. And it sort of goes back to that idea of horses for courses. A polar bear, fantastic hunter in the Arctic. If I drop him in the Australian outback, he's probably dead within 2 weeks. However, if I drop in 50 different species into the outback, maybe 40 will die, but 10 will proliferate and grow. And we have that same idea about how we want to stimulate soil microbes, how we want to enrich the microbes. So these products, or in particular Populate, is designed to specifically do that. So we've identified all of these beneficial functions that you saw on one of the earlier slides. So the question was, how do we harness those functions? How do we screen for the microbes to actually be able to isolate them? Can we make a product to incorporate all of these so you get the benefits? And how do we ensure that this product works on different plants and with different soil types? And can we really get the diversity that we require? And that really started a project many years ago on how we came up with a product like Populate. That's just a little picture of the inside of an autoclave if anyone's interested. So in order to be able to make a product like this, we obviously need quite a lot of equipment. So what happens is we get soil samples in, we freeze them in a -80 freezer, very similar to the freezers that people might be familiar with from the COVID vaccination clinics, exactly the same thing, -80. We have incubators that allow us to grow microbes in controlled conditions. We have biohazard hoods, so in order to be able to grow things in a safe manner, and also in order to be able to keep things sterile and nice and clean, we often work inside a hood. And we have an autoclave so we can actually make that media. So on these slides, you see a few of the methods we have for growing. One of them is to grow things on agar plates. I'm not sure if you're familiar with agar plates, but they're literally, excuse me, a solid growing media that contains that basically contains nutrients on which we can grow things like trichodermas and penicilliums. On the other side, you see a shaking flask where we grow things in a liquid media. Once we've isolated those microbes, we then store the individual beneficial microbes in little trays, again at our -80, or on the other side, you see we can also freeze dry these microbes. The advantage of freeze drying is we can store them in an ambient environment, so you don't have to store them at -80. And the other thing is they will last in a freeze-dried state for up to 50 years. Within a -80, you probably can expect to get about 20 years of storage out of it. So the idea is to have effectively a seed bank, which we can go back to and in effect, take those beneficial microbes out and regrow them. So that's the picture I showed you before. So let's go through the workflow of what we do in order to get microbes. So the soil comes in from gardens and farms around Australia. We dilute that in sterile saline. We plate that onto the selective media. So in some cases, that may be media that shows up the ability to solubilize things like potassium or phosphate. On one side, you see the sort of diversity that we get. So you can see on that plate, there is some fungi, so that fluffy white material. You see a lot of individual microbes, different sizes. So we isolate all the different ones. So, you know, we look for different shapes, different colors, different sizes, different growing habits, different smells. So they all get isolated individually. But occasionally you also actually see something which is quite distinct. And that's what you see on the other side there, on that plate that's labeled with K. That's insoluble potassium on that plate. And you see that one microbe, there's a clear zone around one of those microbes. What that microbe is doing, it's solubilizing potassium. So it's actually taking potassium, in this case trapped in mica, and it's solubilizing it and making it available for plants to use. So we have to remember that plants can't use nutrients unless they're in a soluble format, okay? And so potassium, and in particular phosphate, are things that are locked up very, very tightly, and they're often not available for plants to use. So here you see another set of plates. On one side, you see that potassium plate. You can see one microbe is very good at solubilizing it and the other one doesn't. That red plate that you see That is a plate looking at the ability to solubilize or break down cellulose. So one of the key elements in nutrient cycling is to actually be able to break down organic material. And a lot of organic material consists of cellulose and cellulose-like materials. So if you think about your thatch in your lawn or the stubble in the field, that's mainly cellulose. So the idea here is to isolate microbes that can break down cellulose. If you have them, you can then release the nutrients trapped within that organic material. So we look for those. So the ones that have that orange halo, they're capable of breaking down the cellulose. And the one that you see there called 1D or ID is the one that can't break down the cellulose, so the plate stays red. We also use somewhat more high-tech methodologies where we specifically look for things like the ability to produce growth factors. So in this case, what we do is we grow the microbes up in a special solution. Then what we do is we really measure the amount of growth factor those microbes are able to produce in that solution. So on one side, you see a thing called a spectrophotometer, and you see all those little tubes. Those little pink colors in those tubes are really the ability for microbes to produce different levels of these growth hormones. In this case, we're looking specifically at auxins. The critical thing that I spoke about before was the ability to suppress or inhibit the growth of pathogens. So what we do is every single microbe we get in, we then run through a bank of pathogens and we have a look to see which ones are capable of stopping their growth. So there you see a plate with all the different pathogens we have. We have about, at the moment, about 28 different pathogens that we got in from the Department of Primary Industry in New South Wales. And some of them are really common pathogens that people would have known about, things like Pythium and Phytophthora. I should point out Pythium and Phytophthora are not truly fungi, they're oomycetes. And then some that people may not be as familiar with, things like Fusarium or Rhizoctonia, which are really problems for vegetable growers and people that are growing cereals and that type of thing. We're currently doing a couple of projects specifically looking at black dot in apples and also on hull rot in almonds. So it's not only that we isolate these things, we actually then prepare specialized mixes for people to be able to trial on their farms in order to see can they suppress a disease for which they're currently having to rely upon a chemical, which is often difficult to use and can be damaging to other life forms on your farm or property. So we have to screen a lot of individual microbes, and often you'll screen 50 or 60 and you'll only end up with one that does what you want to do. So it is quite a laborious way of doing things, but it's just part of what we do in the lab. So you'll see here a few slides, a couple of slides, and often you'll see sort of areas where the fungi is growing. And you see other areas where the fungi can't grow. And there'll be some more slides coming up in just a minute that shows that much more clearly. So most people are probably familiar with go-go juice, and now we have Populate. So go-go juice was really the first foray of Neutrog into this area, and we started that about 15 years ago. And just because we've brought out Populate, people shouldn't think that we've dropped go-go juice. Go-go juice is still continuing to be developed. Okay, we're not dropping it off. We're actually continuing to make go-go juice better. So the advanced formulation of go-go juice, which is really now the standard that you get, has 10 of those beneficial microbes we found from our screening process in it. So it's still a great product. But for Populate, we've effectively taken 42 of those beneficial microbes and made that into a new product. So the primary role of go-go juice has always been to stimulate nutrient turnover, breakdown of organic material, to feed both the plants and the microbes. Populate is really targeted for a wider variety of beneficial roles. So, increasing things like disease suppression and the production of plant growth hormones. So that just gives you a little bit of an idea where those two products are relative to one another. A lot of people ask me, what's the difference between Populate and go-go juice? And I often say to them, what's the difference between Arnold Schwarzenegger and myself? So, and I'm the go-go juice, obviously. So let me now talk more about Populate specifically. So Populate contains this high number and wide diversity of these really specific microbes. And And these microbes have really been chosen to cover that range of function that we spoke about. So on those plates here, you can see what I call the angel of death on one side. It's got like these black wings. That's actually a pathogen that you find affects a lot of potato growers. It's called black spot, and it's black dot, sorry. And what it does, it causes a slight grey scuffing on the surface of the potato, and that potato is then rejected. Now you can see the inhibitor C1 is very, very good at stopping the growth of that pathogen. It will not grow anywhere near it. Similarly, on the other side is a rhizoctonia, which if people are familiar with cereal crops, is a major issue in cereal crops. Again, there's a pathogen there that inhibits the growth of that. Inhibitor stops the growth of that pathogen. Whereas there's another one on there called I5, inhibitor 5, very imaginative name, I know, which doesn't specifically inhibit rhizoctonia. And here's a range of others. So we have some that will inhibit Pythium. So a lot of people have Pythium in their lawns. It's a major problem. We actually have one of the largest turf growers in Australia who use a combination of these inhibitors on their turf farm. What we do is we spray the pellets that they use with these inhibitors, and then they put it out, and it reduces the level of Pythium. So you can see we cover a range of different things. So Pythium, Phytophthora, we've had some good results with Phytophthora with growers in New South Wales, and any of you who are signed in or listening in from New South Wales understand that, you know, with the amount of rain you've had, things like Phytophthora, which love water and move through water or using water through the soil, that an inhibitor for that is actually a bit of a game changer. So we've had some really good results with that in trials in Australian natives, so tea trees and that type of thing. Sclerotinia is another one, which if you're growing vegetables is a major pathogen. Again, we have inhibitors for. And lastly, but not least, is that plate there on the bottom that shows the ability to solubilize potassium. I want to just give you a few examples of what we've seen when we've trialed this. Now, these trials were done by elders, and what we're really looking at is what advantage does Populate give you if you're using it, say as a grower, but also if you're using it in your home garden. You can use that on your vegetable crop or on your tree crop or even on your lawn. So you quite clearly see that the control on one side isn't as healthy. It's not as lush, it's not as green. Versus Populate, you can see the plants are much healthier. This is really an example of what we see when we look at the harvestable products. You can see the Populate capsicums are much bigger. They're healthier looking compared to the untreated control. This is a commercial grower for tomatoes, quite obviously, and you know, to the untrained eye, including myself, I'm not trained in this area. That looks like a fantastic crop. You know, most people would say that's a fantastic crop of tomatoes. But you compare that to the treated side. You can see the tomatoes are bigger, but what's really important is the plants are healthier. The stems are thicker, meaning they can carry more nutrients and they can actually support more plants, okay? Or more, in this case, tomatoes or fruit. If we look at the quality of the quality of fruit is much better when you use Populate. Now, the explanation for this is actually fairly simple. If you think about a plant is normally under stress, okay? If all animals or plants suffer a certain level of stress, if you can reduce that level of stress, say it's disease or the ability to gain nutrients, then the plant has the ability to use that additional energy for production of fruits or flowers or growth. So a non-stressed plant is always going to give you better production. And the whole idea behind things like applying Populate is you reduce stresses. You reduce stress like, for example, the pathogen load in the soil or the ability to obtain nutrients. And then we've seen some really good results that Populate can potentially also help with things like frost stress. So what we call abiotic stresses. So these are stresses that aren't caused by biology, but are caused by the environment. Okay, so reducing stress will give you better plants and, in effect, better outcomes. Here's another example for turnips, parsnips, sorry. Parsnips, of course. You can see the untreated ones are much smaller, much shorter. The treated ones are much longer, and that's an indication that nutrients actually, that deeper down in the soil, that plant chases with its root, chases the nutrients. So it means that in the Populate, you're getting better nutrient cycling. You're getting nutrients throughout the entire soil profile, which means the plant chases those nutrients and has access to it. Okay, so there's a lot of positives that come out of using Populate in that respect. Lastly, but again, not least, this is an example of what happens when we apply Populate to turf. You can see on day 1, there's quite a bit of damage on that turf. Within 3 days to 5 days, you actually see a remarkable recovery in your turf. Now, we've got another turf grower in New South Wales, one of the largest ones there. They're now applying Populate regularly on all their turf, simply because what they're seeing is they're seeing much better growth and they're seeing less stress in their plants. So if you want to use Populate, there's a couple of things we need to consider. Okay, now the microbes in Populate are in effect in a state of stasis. They've been put to sleep. So what you need to do is you need to dilute them out, and that's really, really simple. In fact, you can just use 40 mils of Populate, add it to a standard 9 to 10 litre watering can, and then simply water around the drip zone of your plants. Okay. Now, a lot of people have started using Populate as a soil drench prior to putting in new plants. And that's a really good idea, because what you're doing then is you're getting those microbes that produce those plant growth hormones, which are involved in root development, exactly where you need them. You put them around the root. So you can do that simply by soaking your plants in pots, or if you've got bare rooted plants, simply soaking them in a diluted solution of Populate. Now, you can use it in the soil, which is what we really designed it for, but potentially there are uses for foliar applications for disease suppression. It's not the general way we use it, and the reason we don't generally use it as a foliar spray is that all microbes that are isolated from the soil tend to be sensitive to UV light. So if you do want to use it as a foliar application, you can. Just consider that you should really be applying it in the evenings or on the underside of leaves where they're not exposed to the direct sun. The same goes for actually putting Populate out on your garden. Put it out in the evening, put it out on a cloudy day, or perfect time to put it out is maybe when you're expecting some rain, or if you're going to water it afterwards. Because what you want to do is you just want to get those microbes away from the soil surface, away from where they potentially get dried out, or where they're exposed to high levels of UV. Okay, so when should you use Populate? You really should use Populate whenever you've had a major change in the environmental conditions. So we're talking about things like flooding events. When your soil floods and becomes waterlogged, a lot of the beneficial microbes die. They just can't live in those anaerobic conditions. So that's an ideal time to put Populate on. If you've had a really hot, dry spell for a week or 5 days or longer, put Populate back out. Again, drying kills a lot of the microbes, okay? Some will survive, but others will get killed by those conditions. The change of seasons between summer and winter, obviously here you've got a major change. You're going from cooler conditions to warmer conditions. So again, some microbes love the warm, some love the cold. So again, if you reapply, you'll get that re-inoculation effect. Vegetable gardens, pretty much whenever you're changing your crops. So if you're putting in 4 crops a year, summer, spring, winter, autumn, by all means, between each of those, it's a good idea just to put some Populate out, because not only is there selection of microbes in the soil, but the plants also select certain microbes they want to interact with. And so the idea here is that if you're putting a different crop, the previous crop may have selected out microbes that the new crop doesn't necessarily want to interact with. So again, if you Populate out, you've got a better chance of doing that. It's safe. You can use it on your indoor plants, your outdoor plants, on your orchids, on your African violets, on your ferns. It's a perfectly safe product. It won't burn. It's not a fertilizer. It doesn't have a high nutrient load. The idea behind this product is it acts as a probiotic. It acts as a way of, in effect, giving your plants a protection and insurance policy. So think about it as I've got those microbes there and they're protecting my plants year round, okay? Now, 1 litre of Populate, once diluted, you'll get 1,000 to 2,000 square meters of your garden. So for most people, 1 bottle of Populate will do your entire garden. And that's the rate that we actually also recommend as put out as a commercial setting. So the idea is buy a bottle, use it. It's got quite a long lifespan on it. So if you've got a smaller garden, you can use it for a year, 2 years. It doesn't really go off, okay? And lastly, but perhaps not least, just remember these products can only work if your plants are actually growing and are active. If you just put them on the soil and it's the middle of winter and all your plants are asleep, it probably won't work as effectively as, say, in spring or in autumn when some plants are really taking off. If you got your bottle of Populate, I tend to store it in the dark. You know, I put it in the shed somewhere, or even better in the garage, because in your garage it's less likely to be exposed to really high temperature. I know some sheds will get up to 60 degrees inside. So store it in the dark in the shed, or, you know, if you want to, under your laundry sink, which is probably the ideal place to store it. Okay, so I think we're pretty much there. And that last plate there, again, you see a pathogen growing in the middle and inhibitors top and bottom inhibiting its growth. I'll leave it there, and happy to take some questions. Over to you, Helen.
Thank you, Uwe. We have a number of questions to get through. So as we said at the start, we will do our best to get through as many as possible. So I'm just going to select a range of questions. For those who we don't get to, we'll certainly respond via email. And we've actually had a question asked during the presentation about, will the webinar be made available on our website? And yes, it will. So you'll be able to revisit the website. Uwe, David asks us, are you concerned about releasing the cane toad equivalent microbe into an area that would not usually have that species present?
Yeah, that's a very good question. So,
yeah.
So one of the things that we do is we actually sequence and determine the sequence of the microbes. So we determine the genome sequence, and one of the things we do then is we look to make sure that we're not releasing any potential pathogens into environments where they wouldn't be. So I didn't put it on the slide because we are limited a little bit on time, but effectively what we do is we send away our microbes, we get the genome sequence done on those, and from that we can determine whether or not those types of microbes would already be present in other locations. So yeah, the chance of releasing a cane toad-like microbe is highly unlikely. Plus all of these microbes do come from Australian soils, and there's already significant movement of soil material between states if you consider plants from Queensland ending up in WA and trucks moving around. So I'm not really that concerned about that.
Thank you, Uwe. Con from the Gold Coast would like to know, will Populate kill or eject root nematodes?
No, okay, yes. So no, it won't, because we don't know. The best way to handle root nematodes is actually to encourage soil fungi to grow. There's a lot of soil fungi that will actually eat and destroy nematodes. Again, for brevity, I didn't put that video up. So the idea here is two things for root nematodes. Encourage fungi, and the way you encourage fungi is literally by putting out compost and mulches, organic mulches. And lastly, don't overuse nitrogen. If you put a lot of nitrogen out, what that encourages the fungi to do is to actually not kill the nematode. The reason the fungi in the soil kill nematodes is to get at the nitrogen that the nematode contains. So nematode's a little bag of protein, and the fungi effectively looks up on that and goes, oh, here's a source of nitrogen. If you put a lot of nitrogen out, especially in the form of very soluble, high soluble nitrogen, the fungi will in fact not kill those nematodes. It's literally like saying to the fungi, I've got Uber Eats coming every day. I'm not going to bother cooking in my kitchen.
Thank you, Uwe. There is a question that I can answer, which I'm sure our attendees are interested in. I have been asked, where can I purchase Populate? Populate will be available at all of your independent retailers in a 1 litre bottle. It will also be made available shortly in the Mitre 10 network and the Home Hardware network nationally, so you'll be able to access that very shortly. Uwe, another question for you. Julie would like to know, is there an advantage to adding Populate to my compost bin to assist with the breakdown of the plant material?
Yeah. Look, as much as I'd love people to buy Populate to put in their compost bin, I would say you're just as well off using Go-Go Juice for that, because Go-Go Juice has been designed specifically to help in that organic cycling, whereas Populate has been designed really to cover some of the other functions that aren't as well covered by Go-Go Juice. So Populate is about plant growth, nutrient cycling, liberating phosphate, potassium, disease suppression. Go-Go Juice is a really great product, and it really works well in stimulating your compost breakdown. If I could just make one very quick comment about compost, a couple of things to consider. One is, how moist is your compost? Just squeeze it. If your hand comes out moist, it's wet enough. If your compost isn't breaking down and it's wet enough, then invariably it's a lack of nitrogen. So I suggest you go and get a couple of handfuls of Rooster Booster or Bounce Back and throw that into your compost. That will really kickstart it along, because in most cases what the problem is that people don't have enough nitrogen to break down the carbon that they put in.
Mm-hmm. Thank you, Uwe. Robert would like to know, should I use Go-Go Juice with Populate, and if so, can they be mixed?
They can certainly be mixed, but in effect, you'll get all the benefits of Go-Go Juice in Populate. So, you know, you can just use Populate on its own, and it'll work just as well as a combination of the two. The thing that we have to remember is when we're using these type of inoculants, they're not like chemistry where you need a certain concentration to do the job. What you need for these inoculants, you need a certain level of diversity. And then what happens is that you put those out and those microbes multiply in your soil. So unlike a chemical which you put out and it's there and it doesn't grow, these microbes actually grow in your soil. So if you've got the right ones there and you feed them, they grow. So you don't need to use Populate and Go-Go Juice. You can use either Populate or Go-Go Juice. Obviously with Populate you get some additional benefits.
Uwe, Mari would like to know, would you recommend using this in a nursery setting, i.e. watering in after potting up, or just as a soil drench for pots before planting out? Okay, so in a nursery setting, the advantage is obviously if you take advantage of the stimulating root growth, then your plants will establish quicker in a nursery setting. So yes, it's an ideal thing to use in the nursery. And again, when you plant out a plant after you've bought it at the nursery, again, putting Populate in is really ideal, because what you're trying to do is you're trying to ensure that you're suppressing any potential disease that may be in the ground before you start. But also, again, once you transplant a plant out of the pot, what you're really trying to do is you're trying to get it up and going. You want to get the roots developing, and you want to be able that plant to access the nutrients as quickly as possible. Yep. Lovely, thank you. Ruth has a question which I think probably a lot of people would like to know. She says, I assume one can use sea mungus with Populate. Is this necessary?
Okay. So you're talking about, I'm assuming sea mungus liquid or sea mungus pellets?
I would think probably pellets is what Ruth is.
Okay, yeah. So it's interesting you should say that, because I only gave a talk to the Rhododendron Society last week, and people are swearing by the combination of sea mungus pellets and Populate. They're absolutely saying it's a miracle. So the thing about the sea mungus pellets is they do have a lot more nutrients in it. So you're still feeding your plants. And sea mungus pellets are really what I use in my garden for 80% of the time. And I love sea mungus. It's a great product. So yes, you can use them together, because the thing is that when we have a pelletized product, obviously that product is quite dry, and some of the microbes that we would like to have don't really survive that well on a dried product. So by using the combination of Populate with sea mungus pellets, you'll actually get a double whammy. You'll get the nutrients from the sea mungus, you'll get the benefits of the kelp and the humates, plus you'll get that biology, the additional biology from the Populate.
Yeah, that's fantastic. Kelvin has asked, and Kelvin, I'm glad you're enjoying the presentation, and thank you for the comment. He would like to know, have we done any work in managing brown rot in stone fruit?
Not yet. It is one of the things that we're looking at.
Yes.
The reason we've chosen this set of the set of fungi that we have is that they're soil borne, most of those, and the other reason is that we can actually grow them in the lab. So if we can't grow them in the lab, then we have to grow things on living plants, and we got a lot of projects on the go, and that project of using living plants in a tissue culture system isn't something that we've started yet, but it's certainly something we're looking at, because I have had quite a few inquiries from people that want us to look at specific diseases which aren't so easily done just in a laboratory setting. But yes, it is something that we'll be looking at.
Yeah. And Maria would like to know, is Populate suitable for use in wicking beds in my backyard veggie patch?
Yes, it is. You can use it there. It's not a problem at all. You can use it in your wicking beds. You can use it if you're doing hydroponic growth. It can be used pretty much for anything. For hydroponics, I would say if you're using a UV or an ozone sterilizer, obviously you're not going to get the benefits of Populate. But for wicking beds, yes, you can use it very easily. Just going back to your previous question from Kelvin, the thing that we have noticed is that the microbes that we do have in Populate, the inhibitors, have quite a broad spectrum. So there's a good chance it will work against brown rot. We just haven't done the trials or the experiments yet.
Yeah. Thank you, Uwe. Julie would like to know, or she comments, I store my go-go juice in a small garden shed which gets very hot in summer. Does this cause deterioration of the product? Yeah,
so that's one of the things that is a bit of a problem. Having said that, we got a sample of go-go juice back from 2011, which we tested in 2019. So it had been sitting in a shed for 8 years, and it still had 80% of its original activity. So you do lose some microbes. Some of the microbes are more sensitive to extreme temperatures. I would say if your shed gets really hot, let's say 55 to 60 degrees inside, you have probably cooked the product. Having said that, there are a range of microbes. So the ones that form spores or form endospores, so some of the fungi, some of the bacillus species, they will survive extremely high temperatures. You can literally almost boil them, and they're still viable, because they form what you would consider almost a seed inside the parent. And so although the parent microbe dies, the spores survive. Yeah. But yes, you do lose some activity.
Yeah. Thank you, Uwe. Sean comments or asks, would aerating a dilution improve the application?
Right. So it really won't, unless you're providing an additional food source. So if you want to do that and you want to put Populate in, you can do that. You just have to remember that there's competition, and if you're going to brew it, there'll be competition in that liquid, and that competition may be different to the competition in the soil, and so you may actually be, killing or reducing the number of some of the beneficial microbes you want in your soil, because you first conditioned them to a moist environment. So there is that thing where, yes, you can get more numbers, but you're actually doing a selection in an aquatic environment, and then after that you're doing a second selection in a soil environment, and they can be quite different from a nutrient perspective, a pH perspective, an oxygen perspective. So I generally would say just put it in your watering can and put it out, and that's great. Or what you can do is, some people are looking at using those spray packs. They're ready to use spray packs that we can get for go-go juice. What you do there is you basically dilute the Populate 1 to 4 with water, put it in your spray pack and spray it out. So if you've got a large area like a lawn, obviously being able to put it out in a spray pack like that is
much easier. Uwe, Graham has a terrific question, and he says he represents many vegetable growers in Botswana. What are the optimal soil pH and temperature conditions for Populate, And is Populate a one-off application, so to inoculate the soil, or are multiple applications required?
Right. So the idea behind Populate is that there is no optimal soil. So the whole idea was we're trying to isolate microbes from as diverse environments as possible. So because the thing is that we want to be able to use Populate on pretty much all soil types. So if you look at Populate, it has about 300 to 400 different microbes in it. So it has a base of about 250 to 350 microbes. On top, we add these additional 40 that we specifically selected. Now, if you put that out in a soil with a pH of 8, there'll be a certain select group that will love that and they will live there. If you put it out in a pH of 6, there'll be a completely different group that will be there, because that's all about the diversity you find in the soil anyway. So there isn't an ideal soil that it works for. Okay, the idea is that it's supposed to work across as many soil types as possible, because that's how we select the microbes. To his second part of the question, what we'd like to think about is that we like to think of this as being a real probiotic, in that it actually contains microbes that are naturally found in the soil, and their numbers and their diversity varies with environmental and seasonal conditions. So it's not something that you would put on every day or every fortnight. It's not like a lot of these sort of probiotics that are sold to the humans who take one every day or one every week. The idea behind Populate is you put it on, and only when you've had environmental changes, so whether that's weather conditions or change in the crop, would you reapply. Okay, so it's not a one-off. It's not a one-off and it lasts forever, but it's a sort of thing that you might apply 2 to 4 times a year.
Yep. Uwe, look, we have had so many more questions to get through, and they're all really fantastic questions, but we've really run out of time. Before I pass back to Zell, I would just like to say to everybody out there, the product will be available at your independent retailers. We would encourage you to ask them for it, because we get around to many of them, but it doesn't hurt if you go in there asking for the product. And very shortly will be available in the Mitre 10 home hardware network nationally. So Uwe, thanks so much. You've been absolutely fantastic. We will get back to everybody who we didn't answer the questions for in the webinar, and I'll hand back to Hazel. Thank you.
Thank you very much.
Thanks, everyone. We just wanted to thank you all for taking the effort to listen, and I always learn something when I listen to Uwe every single time. And I think that this has been, just looking at the number of questions specifically, I think this points to the appetite to hear more from Uwe in the future. So I think it's been a great way for us to understand how much interest there is in our products and understanding what biological fertilizers are all about. So thank you all for attending, and we'll look forward to doing it again soon.